With audio. Hello, hello. Hi, everybody. 52 degrees in Burlington. Feeling like spring. It's not spring. Don't think it's spring. You'll only get sucked. You'll get lulled into this false sense of security. And in a week, it'll snow 3 feet. So don't get sucked into it. Enjoy it, but don't believe it. Don't believe it. Okay, what do we got? 32. That's 3/5 or 60%. Wait a couple minutes. Kaczynski and Kaczynski. Kaczynski. Ariana Dupre. Ariana Dupre. Are you related to Ariana Grande? Or does it not work that way? I guess it doesn't work that way. I'm sorry. Are you at least a fan of Ariana Grande?
Yeah, a little bit.
And Ariana.
Ariana Coolidge is over there too. Name incorrectly though, because you're right. She's wrong. This is not your fault, Ms. Coolidge. I don't know why, but I just feel the need to make fun of your names when I'm looking at them here. It's fun. It's fun making fun of people. Christy Trask, always on task. That was terrible.
Always a comedian.
Always trying to be a comedian, not always succeeding. So you can always tell when I'm on my computer and when I'm on my wife's computer, because I've got the background changed, the virtual background, and also my camera really stinks. This camera's pretty good, so looks a lot better. Oh, and I haven't teased this person in a while. Hey, Robin Russell, how you doing? Good, good. Okay. I joke. All right. Well, why don't we just do this? We're finishing off chapter 12. This particular lecture, I guess, like all the other ones, may be a little bit short because there's not much left to this chapter. But we'll finish this off. And also, if you haven't noticed in Moodle, I put game number 2 up. So once this class is over, well, it doesn't open till noon, assuming that this class will be over by noon. But once that happens, you'll have all the information you need to do that game. So I would strongly encourage you, A, to do it because it's worth bonus points, and B, to do it over and over again to get as many points as you can. I did it twice and I got 8 bonus points. I'm sure if I kept trying, I'd get 9, but I'm not using my anyway. I guess if I got 9 bonus points, I could give them out to students, but I'm kind of a jerk, so I probably wouldn't anyway. Okay, rock and roll. I like the way Ashley Goldstein is always smiling at me. Hi, Ashley.
Hi. I'm always so positive.
You are. I love that.
Thank you.
Okay, here's my PowerPoint. So what we're going to be talking about today is start out, well, 2 things really. One is targeting. How does the cell get things to where it needs to go? And the other is exocytosis and endocytosis. So targeting, you know, whenever we talk about things inside the cell, me especially, you try and make a story out of it so it makes more sense. I mean, our brains have evolved to learn things in terms of a story or a process. And so, like, I'll say something like, and then this protein wants to go to the rough ER, so then it moves backwards from the Golgi to the rough ER where it feels more comfortable. It's all BS, you know. It has nothing to do with the protein having intention. Proteins don't think, believe me. Oh, never say believe me, by the way, especially since our last president. Every time he'd say believe me, you knew he was lying. I'm not lying though. So anyway, how do things get to where they need to go? It has to do with tags that are put on the proteins or the vesicles, and then in the cell that read that tag and needs to go. And one of the things I'm leaving out here, talked about it last, is when vesicles are moving, remember, it's always a molecular motor, kinesin or dynein put on that vesicle, and the motor put on a microtubule track, and it's pulling that vesicle where it needs to go. So that's what all the movement's about. And I leave it out because, I mean, come on, how much can you say in one day? So anyway, let's get to the meat of the heart of the matter here. So there are two main things that are going to be tags here. One is a tag on the protein itself, and the other is a tag on the vesicle. So either can be used to move things to where they need to go. In terms of on the protein, there are I think three different aspects of the protein that will determine where it goes. And they're all based on the amino acid sequence of that protein, which makes sense, right? The gene has got all the instructions for what that protein needs to do, where it needs to go. So the primary amino acid sequence then, in the primary amino acid sequence, there are, short sequences that tell these proteins, or that decide where these proteins are going to go. We call these tags, basically. And so they're about 3 to 5 amino acids long. The part of the primary structure, so it's not something that's added to it. It's right there in the protein. And what these do is, you've got like 3 amino acids sticking out somewhere in the protein, usually on the luminal side of the rough ER, and there'll be a receptor, a big protein receptor in the rough ER that will grab that, and it's going to hold on to it. Now, this receptor could stay in the rough ER, which means that protein is going to stay in the rough ER, or this receptor could move to the next compartment, the Golgi, and take that protein with it. So that's how trafficking happens through tags. The tag binds to a receptor, and then the receptor moves around. What kind of tags do we have? We've got 2 different types of tags. One is called a retention tag. So this will bind to a receptor that's going to stay in that compartment, and it'll hold that protein in the compartment until it's ready to leave. So our example of a retention tag is RXR. It's not on this slide. Oh no, that's the rough endoplasmic reticulum, for example. So right here, here's your or my ultrasorbent calcium channel. Let me back up a tad, because I talked about this earlier in the class. Oh, here it is. Slide 16. Here's your Voltspin calcium channel. Now, it doesn't operate by itself. It's got these other 4 subunits that need to be attached to it before it's going to be fully functional. So when this is made in the rough ER, it's going to be, it has a retention tag on it that will bind to a receptor in the rough ER. And notice here's your beta subunit of the alpha receptor here, attached to it on the luminal side in the rough ER. Let's take a look at that in a later slide, which is here, slide 35. Okay, so here's that alpha subunit again. And in the rough ER, so this would be the lumen of the rough ER, that little cytoplasmic loop comes out and it binds to this. This is the receptor in the rough ER that's going to hold it. So it holds it there. Well, eventually it's going to have to be released. What causes it to be released? It's being held there in order to finish the assembly. So part of the assembly is hiding that tag. So here's your beta subunit again. Beta subunit then gets bound to that same place that the receptor was binding to. Now the receptor can't bind to the alpha 1 anymore, and it can be released from the rough ER and moved on to the Golgi. Okay, so that's the retention. It's 3 amino acids, and RXR is one example of it, but three amino acids that will bind to a receptor and hold that protein where it needs to be until it's completed. Now, let me back up a tad again, letting you see behind the screen here, behind the curtain of how things really go on. RXR, what does that mean? I keep saying that. I like to be the kind of professor who explains what he's talking about. So RXR, R is arginine, X is any amino acid, and R is arginine. So this is basically an arginine, two arginines spaced out by some other amino acid in the middle. Notice what side chains are on amino acids. This isn't really important for this class. It's more biochemistry, but I think it helps to explain things. Our arginine is a positively charged side, has a positively charged side chain. So that sticks out like a sore thumb. So you can imagine a receptor protein with two glutamates that are negatively charged interacting with that and holding in place with the salt bridge, right? So that's how these tags are working. This retention receptor here has got most likely some negative charges that are grabbing onto these positive charges, and that's keeping it in place. Okay, so that's your retention tag. Another type of tag we have is the retrieval tag. And KDEL is an example of a retrieval tag. So again, K is lysine, positively charged. D is aspartate, negatively charged. E is glutamate, negatively. And L is leucine, which is neutral. Not really neutral, no, it's hydrophobic. So again, you've got positive, negative, negative. You've got some big obnoxious thing sticking out here that's easy to recognize. So some proteins will have that on them, and that will cause them to be retrieved from some forward compartment and go retrograde back to where they should be in the first place. So an example of that is here. This is your rough ER, and you've got proteins that are soluble proteins in the lumen of the rough ER. So enzymes that are operating on proteins as they're being made, chaperone proteins, anything like that, like a soluble protein that's there in the rough ER. So when vesicles, bud off the rough ER, some of those soluble proteins end up in those vesicles. And so they get transported to the Golgi. Now they don't belong in the Golgi, they don't function there. And so the cell then has got these receptors in the cis Golgi that are specific for those retrieval tags. And those receptors sit there until one of these proteins binds to it, and that activates the receptor to retrograde. And they pull that, they go back to the rough ER, and they release the protein back in the rough ER. So that's a retrieval, and it's based on a tag like a KDEL. So again, just going through it again, you've got soluble enzymes here, accidentally get trapped into a vesicle, and that causes them to be moved to the Golgi. They belong in the rough ER, so they bind to these retrieval receptors that then move them back into the rough ER. Okay. So computer issue again, I can tell I'm running out of energy. Hang on one second, I'm just going to get my charger and I'll be back. Okay, I'm back. Okay, so that's our retrieval tag. Any questions or comments on retrieval tags? Okay, so those are two examples of amino acids that are used as tags. Now, also on the proteins can be tagged with glycosylations. It's kind of an example, but it's different. So we talked about core glycosylations that happen in the rough ER. So those glycosylations get added to asparagine residues and linked, and you've got receptors in the rough ER that can grab onto that glycosylation, and that can either be a retention receptor keeping it in the rough ER until it's ready to move on, or it could be retrieval, bringing things back when they've got those glycosylate, the specific sequence of glycosylations on them. So I believe most of your core glycosylations are about that, about targeting and moving things around. And then the glycosylations in the Golgi are more about the immune recognition. So that's another kind of tag that is used to move things around. And again, it's based on the amino acid sequence of the protein, because the protein has got the asparagine in the right place of the protein in order to add that to it. So again, it's all directed by the genes, because it's right there in the amino acid sequence. And then the third thing about proteins that can be targeting is the length of the transmembrane regions. So when we were talking about co-translational insertion, I said that you've got these start transfer sequences and stop transfer sequences. They're all transmembrane regions because they go into that pore, get stuck in the pore, and then get pushed out of the pore and into the membrane. So those transmembrane regions can be anywhere from 18 to 22 amino acids, and that's not random. The reason for that is the membranes differ whether you go when you go from the rough ER out to the plasma membrane. All along the way, they get thicker and thicker. So I talked about this last week too, or maybe it was Tuesday. Uh-oh, shit, what happened? Oh, man. Stop. Okay, hang on a sec. Can you guys all still see me? See me? Ah, okay. Because my computer just skitzed out here. Sorry about that. Okay, so the hell was I talking about? The length of the TMR. So it's not random again. If a protein is supposed to be in the membrane of the rough ER, it'll have a shorter transmembrane region because the plasma membrane there is thinner. It's like 5 nanometers at the rough ER. Plasma membrane is like 8 nanometers. So your longer TMRs are in proteins that are destined to be in the plasma membrane. Okay, so that's another way of targeting a protein to where it needs to go. It's got a transmembrane region that is the right length for that compartment. If you had a 22 amino acid transmembrane region in a thinner membrane like the rough ER, it would be unstable because you'd have some hydrophobic amino acids sticking either in the lumen or in the cytoplasm, and hydrophobics don't like to be that way. So once it gets into the plasma membrane, now it's all totally enclosed in the membrane and it's happy. Or vice versa. If a transmembrane region was short because it's supposed to stay in the rough ER, if it was in the plasma membrane, you'd have hydrophilic amino acids being pulled into the membrane in that hydrophobic environment, and it would be unstable. So that the length of that transmembrane region helps to direct that protein to the right compartment because it's going to be most stable in the right compartment. What else? Okay. So in addition to the proteins having, tags or having ways to know where they need to go, the vesicles also can be tagged. So the phospholipids also have their own mechanisms for directing them to where they need to go. One thing which we've talked about, this bad boy comes up on a regular basis, and that's phosphatidylinositol. It makes up, I'm going to estimate 7% of all the phospholipid in a cell, but it's used in so many different ways for so many different things. We saw it already in a regulation of the cytoskeleton with when it becomes phosphorylated to PIP2, it drags the CAP-Z off and allows polymerization to happen. In chapter 23, we'll talk about calcium release because of PIP2. Here, when you phosphorylate that PI to PIP2, it directs the vesicle to go to the endosomes and eventually lysosomes. Okay, so you're tagging the vesicle by phosphorylating the phosphatidylinositol that's on its cytoplasmic side. Another thing, just like what I said with the length of the transmembrane region, is the thickness of the membrane. So the length of the fatty acid tail will determine the thickness of the membrane, and thicker membranes are going to be directed towards the plasma membrane, thinner ones in the Golgi, and thinner yet in the rough ER. So the length of the fatty acid tail is another way of directing those vesicles. And finally, in terms of the fatty acid tails, there's different levels of unsaturation of those tails. So, you know, making double bonds and causing them to be kinked and stuff like that. So that also can be a mechanism of directing vesicles where they need to go. So you can imagine a system where the protein is directed to the lipid that's got the right thickness for its transmembrane region. And then the vesicle is then directed to the right compartment based on the length of it, the thickness of the membrane itself. Okay, so, and then your book gives a nice example of how this is done, how you deliver things from one compartment to another. And that's here with this MPR. All right, so this is the mannose phosphate receptors. So the enzymes that end up in the lysosome are acid hydrolases. So these are like proteases, carboases, and lipidases, to break down everything that needs to be broken down. And these are acid hydrolases in that they're only active in acidic medium. So if these vesicles were to open somehow and spill their contents out into the cell, they'd be inactive. It wouldn't matter. It wouldn't destroy the cell. And so they need to be directed from the rough ER to the endosome so they can become part of the lysosome. So how they do that is when they are in the Golgi, they get glycosylated. And these glycosylations include mannose, right? Mannose, glucose, and N-acetylglucosamine. So some of these mannose residues on these acid hydrolases can become phosphorylated. Another enzyme in the Golgi is a kinase that will do that. Once it does, it's now a mannose phosphate. It's actually on the 6th acid group, so it's mannose 6-phosphate. You've got receptors then in the trans-Golgi network that will bind that mannose 6-phosphate. These are called MPR, mannose phosphate receptors. So these MPRs then will bind the acid hydrolases and then become sorted into endosomes. These endosomes then will sit around and wait for endocytosed material to come along. And when it does, they will fuse with the endocytosed material, and then several other of these secondary endosomes will fuse together, and eventually it gets big enough that it becomes a lysosome. When it becomes a lysosome, it will acidify itself because it's got a proton pump in its membrane. Remember the V-type pump. And that acidification causes the mannose phosphate to the receptor to let go of that mannose phosphate. And then the phosphate will be removed, and now you've got your acid hydrolase in the lysosome. And then you've got sorting again, where this MPR then comes out of the lysosome and then fuses back onto the trans-Golgi network, and it's ready to grab another acid hydrolase and bring it along. Okay, so that's classic sorting, where you've got a temporary tag on a protein, something that can be changed, and the receptor grabs it, brings it to the compartment it needs to be in, lets it go, and then it gets recycled back to do it again. Beautiful, people. This is what makes cell biology beautiful. Again, what I love about this, why I say it's beautiful, I'm joking around, but why I say it's beautiful is like, it's not magic. These proteins aren't thinking about where they want to go and swimming towards their destination. Everything's everything's coded in that DNA, which gives you the primary sequence of the acid hydrolase, which allows it to be glycosylated. And there are enzymes there that are coded to look for those glycosylations and the phosphate. And it's all just magic. It is magic. Okay, let me shut this off. It's not magic. The whole point is it's not magic. Jeez, I keep contradicting myself.
You know, I heard a few years ago. I heard a quote from someone. Magic is science we don't understand yet.
So true. It's so true. You know, it's like, well, I don't know. I was going to go off on religion, but I won't because I know a lot of people are religious. But yeah, it's kind of, yeah, we don't understand it. It's magic. Once we do. And what was it? There was another quote. It was something like, if technology that's far enough ahead is ahead of a certain population is indistinguishable from magic. People who are more technologically advanced run into people who are less technologically advanced, it seems like magic. Okay, so that's it for targeting. The rest of the lecture then is going to be about exocytosis and endocytosis. So exocytosis. Let us exocytose. Okay, so exocytosis is basically a vesicle with stuff in it is going to fuse with a membrane. Let's say this is the plasma membrane. Could be the Golgi membrane, could be the RFER. It happens on all the membranes. But it fuses with the membrane, and then any proteins in that membrane become part of the membrane of that target membrane. And again, I talked about this last week. Keep in mind the direction of these things. So whatever is in the lumen of the vesicle then becomes extracellular when that vesicle fuses. Okay. And anything that is cytoplasmic will stay cytoplasmic. And then anything soluble inside just gets released from the cell. So this is another figure I like to use talking about that directionality. Here we've got a protein whose N-terminus is in the lumen. It moves its way over to the plasma membrane. It fuses for exocytosis, and now that protein, what was in the lumen is now extracellular. Keep that geography in mind. And then again, this is just regular old exocytosis, where you've got whatever's inside the vesicle gets released. It's a bit of a pop, very, very small pop, and then a sound as the stuff comes out. So we've got two different kinds of exocytosis. One is called constitutive. I use the word constitutive a lot. Constitutive just means it keeps going, it keeps happening. Like there's no, there's nothing controlling it, it just goes. So that's classic for exocrine glands, which are going to secrete out of the body. So something like a pancreas, the inside of your GI tract is not in your body, really. I mean, it's considered part of the outside world. So the pancreas then secretes proteins into a duct, and the duct goes into the GI tract. So that's exocytosis. So those types, and also like milk glands, right? In the mammaries, they make milk proteins and things, put them in vesicles, fuse them into the ducts, and then release out of the body. So those types of things, they tend to be activated to produce the proteins. And when the proteins are made, they're just immediately secreted from the body and they go into some kind of duct or some kind of holding area. Pretty easy, pretty uninteresting. Regulated pathways, a little more interesting. It's where things are made and put into a vesicle, and they go to the membrane and they dock on the membrane and they sit there and wait for a signal. typically that signal is calcium. Calcium increases in the cell, and that causes fusion of the vesicle and then exocytosis of the material. Examples of this are like in your endocrine glands, your say your adrenal medulla, right? Sitting there with vesicles full of epinephrine, just sitting there waiting for that bear to jump out and scare you. And when it does, boom, those vesicles fuse with the membrane and epinephrine gets released and your physiology changes. Also, like nerves, the axon bulbs of nerves are like this, right? The vesicles sitting right at the presynaptic membrane waiting. And when that action potential comes down, the axon voltage hits that membrane, calcium comes in, and the vesicle fuses. So typically it's a calcium message that's causing that regulated release of whatever it is. And here's that nerve example. We talked a little bit about this before. This is like the motor end plate of a skeletal muscle. So you've got vesicles here and they're releasing neurotransmitter, and you've got receptors on the opposite side that respond to it. Now, again, it's not constitutive. They come down and they dock to that membrane. And right next to those docking proteins, some calcium channels are attached. The calcium channels are right there. As soon as that action potential comes, calcium comes in, boom, fusion, and it goes out. How does that happen? I'm glad you asked. So we've got a number of proteins involved in this. Two of them are just for that docking process. You've got a coil tethering complex and a multi-subunit tethering complex. These are just going to grab proteins on the vesicle and hold it in place. And then you've got the business aspect here. You've got the V snare and the T snare. V snare is on the vesicle. T snare is on the target membrane. So you can think of this as the plasma membrane, but it could also be the Golgi or the rough ER or wherever that vesicle is going. And then also attached to the vesicle here is Rab GTPase. So GTPase here, it's going to be breaking down GTP for energy. So this vesicle then gets transported by a molecular motor over to the plasma membrane, let's say plasma membrane, and attaches to these tethering proteins. These then dock. So now you've got your T snare and V snare touching each other. And now it's kind of like primed and ready to go. And then when calcium comes in, the GTPase activates, and this fuses these two proteins together. So they're kind of like touching, and then it's like, boom, now we're in love. Now we're together. And that fusion of the T snare and V snare brings these two membranes together so close that they fuse and then release. And then now you've got your T snare and V snare attached to each other very strongly. And so you need something to get that released. And that's where the SNAP and the NSF comes from. NSF is an ATPase that will use energy to release the V snare from the T snare. And then the V snare can be recycled and used again. And SNAPs are part of that process too. Let's watch a little video. I've got a video that shows this process, and I think it's a pretty good one. So. Working on it, working on it. Okay, hang on a sec. Oh, shoot, that's not what I want. Sorry, guys. Should have had this loaded up before. Okay, so this is just a little 5-minute video. Oh, stop already. Hang on.
We will be discussing vesicle docking and fusion. Proteins destined for many outside the zone must be transported through vesicles. If you saw a video around vesicle building, you know that vesicles carry cargo proteins from the source membrane to a target membrane. After the vesicle separates from the source membrane,
Just so you know, we can't hear any of the voiceover.
You can't hear it at all.
I mean, I can't. I don't know if anyone else can.
Claire, can you hear it? No?
Like going in and out.
There should be a button in the lower left-hand corner where you share the screen that says share audio. Like in Zoom.
Right. I'm going to stop share and then.
Here, but it was very faint.
What was that? Someone else have?
I said I could hear it, but it wasn't very loud.
Okay.
Yeah, you may be able to just turn it up, but I know I was in a conference the other night where somebody was actually able to click a button in Zoom to share the audio of the video they were showing.
Yeah,
And I thought that seemed cool, but I don't know how they did it.
that would be cool.
We might too, while we're watching that might be why.
You know what? How about we do this?
You could just try cranking it up.
Yeah, mine's cranked up as loud as it'll go, but my wife's computer doesn't get very loud. Maybe that's what the problem is. I don't know. But why don't you guys go ahead and click on it's in the notes that I uploaded. So go ahead and look at that yourself. There's no sense wasting your time here if you can't hear it. Okay. Okay, so that is the snare hypothesis. So this is a kind of an up close look at what the snares actually look like. And there's the V snare in blue and the T snare in yellow. And then this is another protein involved in the process, but they wrap around each other really tightly, I should say. All right, hang on a second, let me do something. There was a really cool part of this video I wanted you to see especially. All right, let's just watch it, even if you can't see it, because I think this is pretty cool. Here you do, Dr. Slush. What the hell do you know? I don't know. I'm doing this a long time though. Okay, so let's just watch this fusion. So here you've got your V snare in blue and your T snare here in dark. I guess that's not blue, that's cyan. So that's cyan and blue here. And then this is like synaptobrevin, the purple one, but don't worry about that.
squeezing out any water. Snare filling releases the energy needed to overcome the compression between the two membranes. When the membranes get close enough, hydrophilic heads from each membrane melt.
I guess that's the picture I wanted you to see, this hemifusion. So the V snare and T snare getting together causes this to happen. And this is unstable. So once that's
Causing the closest layers of the two membranes to merge together. Spontaneous flipping is the way to stop vesicle fusion because it forms an unstable hemifused membrane. Because hemifusion is highly unstable, quickly is to form a fully fused membrane. Once the vesicle has fused with its target membrane, the part is released into the luminal side and the membrane proteins in the vesicle become part of the target membrane. After fusion is complete, the vesicular components, such as V snares and vesicular rotating beads, need to be released. Round gaps on the target membrane deactivate vesicular rotating bead to form round GDP, which is released into the cytoplasm. The snares present a more difficult problem.
okay. Oh, yeah, you can't hear it, so I'm not going to do any more of that. But that is your snare hypothesis. Okay. Oh, by the way, here's another mistake. If you see anything with the address facweb.plattsburgh.edu, there isn't a server called that anymore, so you won't get anywhere there. Okay, so that's exocytosis. I kind of think, I'm pretty sure that constitutive exocytosis also goes through snares. Not 100% on that, but I believe that's true. And you know, in today's world, what you believe is all that matters. The truth isn't really that important. Just kidding. But so that's exocytosis. Now this is endocytosis. So we've got a number of different mechanisms of endocytosis. I didn't get through all the notes when I was writing the notes, so I didn't finish this section of the notes. I'll finish it today and upload it again, replace the one that's there. But I've only got through the next couple sections, so just so you know. So endocytosis is basically invaginating the membrane, bringing in stuff, and then making a vesicle inside the cell. So number of reasons a cell could be doing this. It could be trying to like bring something in from the outside, okay, in the lumen of that vesicle. It could be just bringing membrane in, and it, you know, coincidentally brings in stuff from the outside, because those membrane proteins have to be recycled sometimes. Like if you're talking about a axon bulb and a neuron, it's constantly fusing vesicles. Well, you have to bring that plasma, that membrane back in, otherwise you're going to run out of vesicles, and the plasma membrane is going to get huge. So you've got some endocytosis there that's just about recycling membrane and recycling membrane proteins. So that's another aspect of it. Either you're bringing something into the cell or you're recycling lipid membrane. So we've got a number of different types of endocytosis. My personal favorite is receptor-mediated endocytosis. So this is when you're bringing in a specific target. So we'll talk later about penocytosis, which is just bringing in some membrane and whatever happens to be outside the cell. That's non-specific. This receptor-mediated is very specific. It's like LDL is my favorite example. So you probably know about low density lipoproteins. They are the way the liver packages up cholesterol and fat to send it out to the body. So everything you eat, again, goes to your liver processes it into LDL and then sends it out in the form of that LDL. So the LDL is lipid, cholesterol, and also some proteins, apoproteins, that are sticking out from it. So those apoproteins then become ligands that can bind to receptors specific for them, which means, you know, if you're a cell that needs cholesterol, say, let's make you a testicular cell, and you want cholesterol to make some testosterone. So you put receptors for LDL out in the membrane, and then when LDL comes by in the extracellular fluid, it binds to that receptor, and now you can pull it into the cell. So this is how that process works. So let me get a better figure. This is the figure that goes through. We'll go through this piece by piece though. So you've got these receptors in the membrane that are specific for something, and maybe this is our ligand. Let's call this LDL. So LDL binds to that receptor, and when it does bind to the receptor, that causes the receptor to move over into these coated pits. So they're coated, you know, again, you know, before they knew what the hell it was, they could see it on an electron microscope and say, oh, look, that's coated with something. You look at this, you can see this coating on the inside of the plasma membrane, very thick. So this coated pit, what that coating is clathrin. So clathrin then, and also some adapter proteins, are coating the inside of the membrane here. These receptors go and bind to that adapter protein, and then that activates the clathrin to start polymerizing. And as the clathrin polymerizes, its 3D structure is like a soccer ball, basically. So as it polymerizes, it forms a soccer ball, and it pulls the plasma membrane in with it. So this polymerization then of clathrin causes the endocytosis to happen. And then, you know, just like in exocytosis, getting two plasma membranes together is not always easy, right? It's not energy favorable. So you got this other protein, dynamin, and dynamin is going to cause that to pinch off. So boom, it comes inside. And when that happens, then you've got a coated vesicle inside the cell. So we've pulled all these ligands in using the receptors and the clathrin. Once it's inside the cell, step 2 is to uncoat, okay? Take the clathrin off and the adapter proteins. They go back to the membrane and they can be recycled and used again. Now you've got an uncoated vesicle. Now we're going back to something we've talked about already, where this uncoated vesicle will fuse with endosomes, and the endosomes will coalesce into larger bodies, and you end up with a lysosome. So the lysosome then will acidify, and when it does, the ligand comes off the receptor, and then you get sorting of the stuff inside the lysosome. So the receptors will sort in one direction, and they get recycled back to the membrane to be reused. And then what's inside the lysosome can undergo a couple different things. One thing that can happen is it can be taken to the other side of the cell and released, transcytosis, or it could go right into the lysosome or stay in the lysosome, acidify, and break it down. So that's what would happen with your LDL particle. It would be broken down so the cell could then use the cholesterol and fats for whatever it's doing. This is kind of an interesting process, transcytosis. You know, for the longest time, I couldn't figure out, now, why the hell would a cell do that? And like suddenly, bing, a light bulb went off when I found a paper, and it said, you know, how does a mother's milk give antibiotic resistance or no antibodies to the baby? So my thought was always, well, I can understand the antibodies being put into the milk, but once it goes into the child's stomach, it gets broken down like proteins do. But no, it doesn't. When those antibodies go into the small intestine, they get grabbed by antibody receptors that pulls them inside the cell, takes them to the other side, and releases them into the body. Transcytosis. So this is how that immunity gets passed from mother to child, by this transcytosis mechanism. Pretty cool. Okay, so here we are again. Is this different? I don't think so. I think this is the same figure. Any questions about that? Endocytosis, receptor-mediated endocytosis. Another thing that that operates this way is transferrin. I don't know if you ever heard of that, but transferrin is how iron gets into your cells. It doesn't just go in as an ion, it gets bound to a protein called transferrin, and there are transferrin receptors that do receptor-mediated endocytosis as well. It's one of those little bombs I drop, and then later it shows up on exam. It's like, did he talk about that? Yes, he did. Okay, what's this? Oh, this is LDL. Okay, so here's your LDL particles and the apolipoprotein that binds to the receptor. Not sure where I got this or where I added it. I guess there's a new section in your in your textbook that has this in it. Okay, clathrin. Here's our friend clathrin. So this is, you know, a three-sided kind of a thing. And when it polymerizes, it does this or this. So it ends up making these soccer balls, and they're kind of, it's like a soccer ball. So it's round and it pulls the plasma membrane in with it. Here you can see that happening. It's not really flat like this, it's more three-dimensional. And there's a coated pit there on transmission electron microscopy. Oh, and here we go again. You can tell this is one of my favorite topics because it's like, oh, look at that. Look how that happens. So here's clathrin polymerizing and coming in to a ball shape, and that's bringing things in. Okay, I'll stop. Okay, clathrin is a coat protein. Okay, hang on a second. I have to get my notes. So clathrin is a coat protein, but it's not the only coat protein. It's used for bringing things in from the plasma membrane. So it's going to take things from the trans-Golgi network to the plasma membrane and bring things in from the plasma membrane to the endosomes. So we're talking kind of targeting again here like we were earlier in the class. You also have coat protein 1, COP1, and COP1 is involved in retro movement. So it's taking things from the, pulling vesicles off the Golgi so they can be released to the ER. And you've got coat protein 2, which is taking vesicles off the rough ER, uncoating, and then sending them to the Golgi. So, and then caveolin is another plasma membrane thing. It's kind of like a lipid raft or something. I'm not that concerned about it. Don't worry about caveolin. Just know that clathrin now is bringing things in from the plasma membrane. Coat protein 1 is retrograde, and COP2 is anterograde. Oh, there it is. Bum, 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 bum. Okay, so that is, and you also have pinocytosis, which I don't have a figure of here, but pinocytosis, they call it cell sipping. Again, it's endocytosis, but it's not specific. We're not grabbing a particle and bringing it in. You're just bringing a membrane in, and whatever happens to be outside the cell, bringing that in as well. So that's pinocytosis. And then you've got phagocytosis, which is receptor mediated again, but it's a much bigger particle. So this isn't just, you know, a little LDL particle. Here you can see a whole bacterium binding to a pseudopod in a white blood cell, a macrophage or something. And, you know, that binding kind of causes the pseudopod to grow around it, and then you pull the whole thing in, you fuse some endosomes to that, and you digest that bacterium. So that's phagocytosis. And again, the difference between endocytosis and phagocytosis is the size of the particle, much bigger in phagocytosis. And I believe, if I'm not wrong, that's it. That's all we have for this chapter. So, any questions?
So you'll be adding, there were no notes on like the phagocytosis, and
Right.
you'll be adding some of the notes today.
I hope to get it up by 1 o'clock
Okay, all right, awesome.
ASAP. Yep. Okay, any questions? Any other questions? Anyone aware that there's an exam coming up on Tuesday? Okay. Unless it's your birthday, then you don't have to take it, which is BS. I mean, yesterday in my developmental class, someone said, hey, it's my birthday Friday. Can I skip the exam? No, you'll be taking the exam.
I mean, yes, if you want to fail the exam.
Yes, you can skip it all you want. I don't care. It doesn't bother me. I get paid either way, you know? So any questions about the material or content or anything? We got 15 minutes left. We can do a bit of a review if you have any questions. I'll tell you what, I'll stick around. If you want to leave, leave. I'll stick around till 1/4 And if you have any questions, I'll be here for you. Let's wait till all the jerks are gone, then we can talk about them. Tyler's still here.
Did everybody leave? I'm the only one.
No,
That's crazy.
Yeah.
Oh,
we got
So-
24 people.
okay.
Oh,
I had
23
a
people.
question, but it's like, I don't know if the question is cell bio, if it's developmental, because like I confuse the two when the exams close to each other, right?
Okay.
Okay, so I'd ask it like this. So in the exam, there's gonna be like couple essay type questions on if something is knocked out, what would happen?
That
Like-
kind of a thing, yeah.
Yeah. So usually, how do I like go about explaining it? Like, should I just say, okay, well, this not here, so this would happen, or should I like go into more detail? Like,
Well,
because
what you want to say is, this is how the process usually works. This part of the process isn't working, so this is something that could be wrong with it, okay? And usually that last piece, there's several different answers that would be
Yeah.
would be correct. So, but the best way to set up your question is explain how it normally works. And that'll get you 7 out of 10 points. Can't figure out what's that last piece, you know, you got 70% of it anyway.
so you get 7 points just for explaining how it works.
Show me you got a brain in your head. I'll give you 7.
Okay.
Okay, any other questions? Kaczynski's got a question. Her question is, why do you keep picking on me? I just love Polish names. I grew up in a Polish small town. Nicole Lamberton, she has not talked yet this semester. Let's see your face, Nicole. Hello.
How long were you in Poland for?
Oh, no, I grew up in Pennsylvania, but most of the people in the town came from Eastern Europe, like around the turn of the century, 20th century. So
Oh, I get it.
it was like this tiny little town. There were the minorities in our town were the Irish, the Polish, the Italian. Those are the minorities, but other than that, everybody was the same. I think it was like 80% Catholic. We didn't like the Protestants. About human beings, you always have to find someone to dislike. You have to be better than somebody, don't you? Humans suck. Okay, well, I'm gonna let you all go. And
See you, Dr. Slish.
you have any questions, send me an email. We'll figure it out over the, get the game. Later.
Will do.